Yeah. So, in this semester, you have cell biology, molecular biology, genetics, and evolution. So, first we will start with the cell biology. So, first we will look into the introduction of cell biology and the structure of animal cells. So, here all the living organisms, they are made up of cells. So, cells are the basic unit of life. Whether it is animals, plants, human beings, or microorganisms, or whatever it is. So, all the living organisms, they may be very tiny or very large, everything is made up of cell. So, cell is a basic unit of life. So, why we call that as a basic unit? Because the cell does all those functions which a complete organism will be doing. So, that means, for example, if you take any metabolic pathway, whether it is glycolysis, which is very simple one. So to take glycolysis, so every cell will perform glycolysis. Okay, so therefore, that means cell. If you study one cell, you can actually study all the living organisms because living organisms are made up of cells itself. Okay. And we have millions of different species of bacteria, protozoa, plants, animals, okay, which have different morphology, function, behavior, and everything. So we study everything at cellular level first. Okay, they are the building blocks of the life. So here, for a proper definition, a cell may be defined as a structural and functional unit mass of protoplasm consisting of a cell membrane, a nucleus, and a cytoplasm. So, first, any cell, so mostly all the cells will have a plasma membrane, which is the outer membrane, and inside you have nucleus. So, the space in the cell is called as cytoplasm. Okay. Now, the cytology is a branch of science which deals with the study of cell. So that means, so what is the structure, what is the function, what are the morphological differences, everything will be studied under cytology. And now this branch is also called as cell biology. So that is nothing but the biology of the cell. So the study of finer structure of the cell, its components and its physiology, and the mechanism of multiplication, heredity, and development. So all this we study under this cell biology. Is it clear? Now, before going to the animal cell, so first we will see what is the general organization of prokaryotic and eukaryotic cells. So prokaryote means pro means primitive, karyon means nucleus. So the cells which are having very primitive nuclear structure, so that is they come under prokaryotic organisms like bacteria, mostly microorganisms come under prokaryotes. Then eukaryotes, U means true, karyon means nucleus. So true nucleus or well developed cellular structure we can see in higher animals and plants. So those cells are called as eukaryotes. So here first, before that, we have to study about the evolution of cell and cell theory. So according to the theory, that is the living cells have probably originated from Earth by spontaneous aggregation of molecules 3.5 billion years ago. So there was some kind of argument, debate that how a cell was developed. Okay. So this evolution might have started with the formation of organic molecules on the earth, which had no life, which is called as prebiotic life. So bio means life. Before the earth, prebiotic earth means the earth before the origin of life. And the prebiotic conditions on the earth results in volcanic eruptions, lightning, and heavy rains. There was little free oxygen and was no layer of ozone to absorb the UV rays, ultraviolet radiation from the sun. So all these were the primitive conditions. Okay, and this is assumed that the simple organic molecules would be generated in such climatic conditions, and this is called as abiotic origin, that is without any life. Now, amino acids, nucleotides, sugars, fatty acids, organic molecules, all were generated, and ammonia, methane, water, formaldehyde. Then, by the combination, actually, life was originated. So, this is first after abiotic, then pro-life or protobiont was developed. That is, it is the beginning of the life. Now we have all the biomolecules, but there is no life. Okay. Later, during the biological evolution, these molecular aggregates, they organized to form a cell, which is, that time it was pre-cellular, and this is actually the true life. It is called eubiont, okay, which was originated from the protobiont. So here there are three major features. One is the phospholipids and proteins assembled into the cell membrane, which form the boundary of the cell, and development of metabolic pathways to utilize these organic molecules to produce energy. And third is formation of self-replicating nucleic acids like DNA, which are capable of storing this genetic information for the synthesis of proteins. So there were the three major aspects here. So 1.5 billion years ago, actually the first organism of a prokaryote was originated. Then later, multicellular eukaryotic organisms were originated. So this was according to the theory of the scientists. In 1839, even for the origin of cell also, there were some debate. Like some people said, God has created. Okay, some people said spontaneously they arose. Means okay, fine, it is a belief, but when science comes, everything should have some experiments, some results, such kind of things. So scientists have become two groups, a lot of debate, lot of experimentation was done, but they couldn't prove anything. But later, finally, the theory that was approved as a cell theory was given by Sheldon and Schwann. So in 1839, Sheldon suggested that every structural part of a plant was made up of cells or the result of cells. So all the structural components of a plant are made up of cells. And cells has to be born from another cell, within other cells or from outside. Okay. And this crystallization process is no longer, actually he suggested of some crystallization, but that was not there in the final cell theory part. Okay. In 1839, Schwann suggested that plants and animals are composed of cells or the product of cells in their structure. And this actually led to cell theory. So there were three postulates which were suggested by these two scientists. All living organisms are composed of one or more cells. This is first part. Second is the cell is the most basic unit of life, and all cells arise from pre-existing cells. So without any pre-existing cell, there is no new cell that was originated. Okay, these are the final postulates of the cell theory. Now, modern interpretation. Okay, so after this cell theory was developed, lot of research was focused on the cells, and then scientists have identified proteins, nucleic acid, etc., etc. Now, generally accepted parts of the modern cell theory include that all the cells are made, all the organisms are made up of cells, one or more, and living cells arise from pre-existing cells by division. And the cell is the fundamental unit of structure and function in all living organisms. And the activity of an organism depends on independent cells. That means every independent cell is capable of performing some activities. Then energy flow occurs in the cells. So what is this energy flow? That means, so here if you see the biomolecules in biochemistry, suppose carbohydrates or proteins or nucleic acids, anything, so it has to be made inside the body. It doesn't go outside or doesn't comes from outside. Only through food some material will be supplied. But all these processes occur through metabolism, which includes anabolism and catabolism. Here, along with the synthesis or breakdown of the molecules, there is also energy flow, which occurs during this cells, okay, during this process. And then cells contain DNA, which is specifically in the chromosome, and RNA is present in the nucleus and cytoplasm. And all cells are basically have same chemical composition or of similar species. Like most of them, if the species are similar, the composition is also, so only some organisms, there will be some exceptions. Okay. So this modern version of cell theory includes the ideas that energy flow occurs within the cells. Hereditary, that is DNA, will be passed from one cell to other. That is not only from parents to children, like from within body also. Like every time the cells will die and new cells will arise. So from one cell to other cell also, actually DNA will be passed. And all cells have the same basic chemical composition. Okay. So as I told you, based on the presence of nuclear membrane, presence or absence, there are two types, prokaryotic and eukaryotic organisms. So prokaryotic organisms, they lack nuclear membrane and mostly they are simple, primitive organization. They are unicellular, that is single cell will be present, anaerobic, oxygen is not required for their growth. Then heterotrophic, they take up the food or energy from the organic matter. And Haeckel in 1868, he included prokaryotic organisms in the kingdom Monera. Okay. And the smallest prokaryotic cell is pneumonia-like organism. Okay. And the best studied species of bacteria is Mycoplasma gallisepticum. Okay. This causes respiratory disease in poultry, that is chicken and all these things. Now, all of them are made up of lipoprotein membrane, plasma membrane, and the cytoplasm has DNA, which is linear double-stranded. Large granules of ribosomes and small granules of protein are present. And if you see like other bacteria, all of them, they differ in their size, they differ in their shape. Okay, we can see different size, shape, or organization in bacteria. So the rod-shaped bacteria is called bacilli. Spherical-shaped bacteria is called cocci. Spiral-shaped bacteria is called spirula. Okay, if you check the names like bacillus, or like that, lactobacilli.
Lactobacillus,
Yeah. Okay. So in such
Streptococcus.
condition, okay. So in such conditions, we usually see this bacilli. The name indicates rod-shaped structure. Okay. And the plasma membrane surrounding the cytoplasm is three-layered structure of protein. So actually it is, one is lipoprotein layer, then we have cell wall, and in some organisms there is a capsule or slime layer. So usually three layers will be present in the bacteria. And the cytoplasm is granular, which includes free ribosomes, single strands of RNA, granules of proteins, and other mitochondria, chloroplast, endoplasmic reticulum, Golgi apparatus, nuclear membrane, lysosomes, all these will be absent here. Okay. Then only mesosomes are present, and the inner folds of plasma membrane will be having respiratory enzymes. So enzymes that will perform the respiratory functions. And the nuclear material is usually double-stranded coiled DNA without any protein called nucleoid. Okay, so usually in eukaryotes, histone proteins will be present along with the DNA, but here it is absent. And the circular DNA, it measures about 1000 micrometer. And binary fission, so asexual reproduction will be taking place. So it's a binary fission budding will occur. So mitosis it does not occur here. And in flagellated bacteria, flagellum is present, which is a locomotory organ. So it originates within the cytoplasm. So here, prokaryotic blue-green algae cells, they resemble bacterial cells, okay, except that the cells contain photosynthetic lamellae or thylakoids. Blue-green algae, no, they'll have some pigments, which are called phycocyanin, which is blue color pigment, phycoerythrin, which is red color, and chlorophyll, green color, and carotenoids, which are orange color pigments. And this protoplasm is divided into peripheral chromatoplasm and central centroplasm. So chromo means color. So chromatoplasm is a colored substance, whereas centroplasm is not colored, colorless. This centroplasm actually consists of the DNA, which acts as a genetic material. Then in virus, there is no cell formation and is found in the form of particles. So usually we don't consider virus as a living organism because it cannot replicate on its own. It has to depend on other host. So here each viral particle is called virion. And viruses are simple organisms with a circular nucleic acid molecule, whether it is DNA or RNA viruses are also present. And it is surrounded by a protein coat or capsid. Then in animal and plant viruses, the nucleic acid is RNA. While in bacteriophages, that means the viruses which attack the bacteria, it is the DNA which is the genetic material. So this viruses, they depend on another living organisms as a host. So it uses that machinery to replicate its genetic material. Now eukaryotic
Excuse me,
organisms,
ma'am.
sorry?
Excuse me, ma'am.
Yeah.
Ma'am, actually, all these are simple topics now. Can you please go with some other topic? Why? Because
Hmm.
remaining all are little bit difficult. Block 3 and 4.
Which topics?
Block 3.
Block 3.
Yeah, ma'am. Why? Because we are having only 5 days of classes, isn't it?
Oh,
Yeah, that is the reason.
just a second.
Yes, ma'am.
Okay, just a second.
Yes,
Okay. Block 2 also, you are like confident? 1 and 2?
yes, ma'am. I can read it. That is fine.
Means
But
not only you, like, I mean, it is for all the students now.
yeah, remaining maybe why? Because people who have already studied in by PC and inter only will be taking this, isn't it? So
Okay,
they can carry on maybe.
okay.
But
Okay,
this
one
thing
thing
is
we'll do. Yeah, okay. I will just continue with this small. I'll just give a brief introduction about eukaryotic cell. Then I'll
yes,
start the
ma'am.
genetics. Meanwhile, you can just discuss with your classmates and just let me know. Like, because some people means they have that. It is only not you, right? So it
Yes, ma'am.
all
Yes, yes.
classmates. If you have any group, you can just discuss with them. So which all topics you want, you can just tell me. That's fine.
But I don't know how many will respond also, ma'am.
Fine, you ask one. Like,
Yes, ma'am. I'll ask. Okay, today we will discuss this only. Then tomorrow, after I
I
ask,
will
we will
just
continue.
okay. I will complete this eukaryote. Just basically, I'll tell. Then
Yes,
we'll
ma'am.
go to the genetics. Tomorrow,
Okay,
you
ma'am.
just meanwhile you can ask and we will see. Okay.
Yes, ma'am.
Okay, so in the eukaryotic organisms, like as I told you, like we have cell wall will be mostly present only in the plant cells, whereas in animal cells, cell wall will be absent. But other than that, all this plasma membrane, then nucleus, like in the cytoplasm, mitochondria, Golgi apparatus, all these like what you call well-developed organelles will be present. So I'll just discuss this table. That is it, more than enough. Like you can see, the size of the eukaryotic cell will be large, whereas compared to prokaryotic cell. And usually prokaryotic cells will be single cell, whereas eukaryotic cells are multi-cells. Like multicellular means it has many cells. Then cell wall is present in the prokaryotic cell, and it is composed of peptidoglycan or mucopeptide, okay, which is polysaccharide. Whereas in eukaryotic cell, only plants and fungus, they consist of cell wall, and other animals will, this is absent. And this also in plant and fungus also, it is made up of cellulose, which is a polysaccharide. Then nucleus. So nucleus is well-defined. Nucleus is absent in prokaryotic cell. It's called nucleoid, whereas it is well-developed, enveloped in the nuclear membrane, and nuclear pores are also present on the nuclear membrane. And this is seen in the eukaryotic cell. Then DNA is usually circular double-stranded DNA in prokaryotes, whereas it is linear double-stranded DNA in eukaryotes. And mitochondria are absent in prokaryotes and are present in eukaryotes, whose main function is energy production. That is, ATP synthesis takes place, and oxidative phosphorylation, all these will be taken. That is for the energy production. ATP itself is the energy. So that energy molecules are synthesized in the mitochondria. Then ribosomes are present in both prokaryotes and eukaryotes. It is 70S in prokaryotes, 80S in eukaryotes. So 70S means here, so these main S means Swedberg unit. Okay, they are mainly involved in the protein synthesis. Then Golgi apparatus are absent, and endoplasmic reticulum are also absent in the prokaryotes, whereas they are present in eukaryotes. And the main function here is once the protein is synthesized, it has to be packed and it has to be sent to its location. Some proteins are secretory proteins, some are membrane bound proteins, some act as receptors, some act as some enzymes. So in such way that all sorting will be taking place in this endoplasmic reticulum and Golgi apparatus. Then mode of reproduction is mostly asexual in prokaryotes and mostly sexual in eukaryotes. And cell division is by binary fission, conjugation, transformation, transduction in prokaryotes, whereas mitosis in eukaryotes. Then lysosomes and peroxisomes are absent in prokaryotes, then present in eukaryotes, which are the suicidal bags. So they usually remove all the dead cells and debris from the cell. Then chloroplasts, which are absent in the prokaryotes. In some like photosynthetic organism, they are scattered. But in eukaryotes, they are present in plants and algae, but not in animals. Then transcription and translation occurs together in prokaryotes, whereas transcription occurs in nucleus and translation occurs in cytosol in eukaryotes. Then organelles are not membrane bound in prokaryotes, but they are well defined membrane bound in eukaryotes. Replication is by simple origin of replication in prokaryotes, whereas multiple origins of replication are present in eukaryotes. And in the prokaryotes, only one chromosome is present, and that is also not true, which is called plasmid. And whereas chromosomes can vary in number in eukaryotes. Some organisms will have 12 chromosomes, some will have 26, some will have 44, like that. Then examples of prokaryotic cells are archaea and bacteria. Examples of eukaryotic cells are plants and animals. Okay, this covers most of the topics. Still, if you have any doubts, Okay, anything basic doubts also, you can get back to me. So I will now discuss about the genetics. Okay, if you feel any topic is difficult or you cannot follow, you can just discuss and let me know. So I think this is the starting topic. Yeah, I think this Mendel's laws of inheritance is the starting topic of this block. So, what are these Mendel's law of inheritance? First of all, what is genetics? So it's a study of genes. So what are genes, their structure, their functions, everything, how they are inherited, all these comes under this genetics. So Mendel's laws of genetics of inheritance is nothing but Gregor Johann Mendel. He was the scientist. Actually, he studied on peas and he has given some laws. So almost universally, all the genes, majority of them, in all the organisms, they inherit in the way he explained. But there are some exceptions here also. So the first systematic approach of investigation of mechanism of inheritance was done by Gregor Johann Mendel. He was a monk, but he has interest in this subject. So there was no subject at that time, but he wanted to investigate like what happens, like how some plants are tall, how some plants are small and short. And even though if both parental plants are tall, some plants may be short, the progeny. So why all these variations? All this he was interested. Then he did a brilliant and systematic work on garden pea, which is Pisum sativum. So here, after doing some experimentation and calculations, he actually given some principles of inheritance, and that's why he was named as the father of genetics. And whatever the branch that was developed, that was named as genetics. Actually, after his death, after 100 years of his death, actually his these laws, everything were actually um, he published. Actually, whatever the work he has done, he published, but it was there in the library somewhere. But after 100 years, by that time there was no DNA, no information about DNA, protein, RNA. So what is the inheritance material? So at that time some people used to think that protein use is the genetic material. So still there is no clarity. And once the DNA was found as a genetic material, again the structure, then mechanism of replication, transcription, translation. So all these experiments, discoveries, it took almost 100 years, and still some other research is also going on that. But basic information was developed slowly. So after 100 years of his death, like two independent researchers, one from other country and other from I think Australia, I don't remember. Two scientists, they independently they found this paper, whatever he worked on. So they took that paper, they repeated the experiment, and they got same results. So this repeatability is very important in science, because even I do the experiment and you do the experiment, both of us should get same results. Then only that is correct, that theory. So the same thing happened, and both of them actually they published at the same time. Then all his laws, experimentation came into light. By that, till that time there was like nobody knows Mendel, what work he has done and all these things. Then by that time all this basic work was done, DNA, structure of DNA and all that. So then actually genetics branch was evolved, and he was named as the father of genetics. So here, so what are the components, entities which are responsible for the inheritance? So they are called as hereditary determinants or factors, and now they are known as genes. Okay. So what Mendel observed was that each plant had two factors, but so he doesn't know whether they are genes or two, nothing. Okay, two factors, that's it. So one from each parent for each character, and each sex has only one determinant or one factor. Okay, now we know that they are called alleles. Now each gene, for example, hair, suppose hairstyle, style in the sense some have straight hair, some have curly hair. So here the type of hair is the one gene X, suppose gene X is coding for the type of hair, and that individual is having X only, but some people may have curly hair, some people may have straight hair. That means both of these factors, they are the two different forms of a single gene. That's why they are called alleles. Now each gene usually have two alleles, but nowadays even some genes will have multiple alleles also. Okay, so alleles they code for one character. Okay, so they are present on the homologous chromosomes. So the chromosomes which are having like same set of genes, and so thus alternative forms of genes are called as alleles. Then gamete is a sexual unit, like whether it is egg or sperm, like that. Okay, and each sex cell has one member of gene, one allele only present. Okay, then contrasting characters. So the individual plant features with marked phenotypes and differences are known as contrasting characters or traits, like red flower, white flower. Contrast completely. They are not shades also, like black and white, complete contrast. Okay, like that. Tall and dwarf. Then dominant and recessive means when a cross is made, usually only one will express, one character will express. Even the other one is present, the other one won't express. Okay, so that type, the one which is expressing, that is called dominant character or dominant gene and dominant allele. And here the one which is not expressed or masked, that is called recessive or character or recessive allele. For example, we have a red flower and white flowered plants and they are crossed. We'll get the progeny all are red flower. That means though there is a character called white here, it is not expressed completely, is dominated by the red color. So that is called red color is dominant, white color is recessive. And red color is represented by capital R and white color is represented by small r. So whatever it is, recessive allele is represented by small letter and dominant allele is represented by capital letter. And Mendel here studied 7 characters in garden pea and recorded the dominant and recessive behavior of all the 7 characters. So all these have this, when he conducted, so all the characters showed similar what you call pattern. So one is dominant gene expresses in F1, that is first filial generation, first generation, whereas recessive gene is masked. Like suppose for your simple, you have to just whatever I'm saying, just imagine. Suppose there is capital T, which is coding for tall, small t, which indicates short. Okay. If you have capital T, so two alleles are present, right? And suppose you name it as like mother or father, like that base. From mother is having capital T, father is having small t, small t. Okay. When they are crossed, that is when the plants, two plants, they are crossed. So this from this capital T, usually one will be passed to the next generation. So anyway, there are two copies of capital T. So one copy of capital T will get. And from other plant, one copy of small t you will get. Now the progeny which is having, it is having a hybrid, that is heterozygous. It is called as capital T from one plant, small t from other. So genetic wise, genotype wise, it is capital T and small t. But phenotype, that means whether it is tall or short, if you see like that, it will be definitely tall, but not short. Why? Because capital T is dominant and small t is recessive. So even though small t is present, it won't get expressed. So this generation is F1, first filial generation. So here only dominant gene expresses, whereas recessive gene is completely masked. Okay. And the dominant gene is represented by capital letter and the recessive gene is represented by small letter. And dominant character is usually wild type, whereas recessive character is usually mutant. So only in some, because there are some mutations or variation between two organisms, so usually wild type will be capital T. So what kind of characters he has studied? So 7 characters. One is plant stature or height, position of flower, shape of the pod, color of the pod, shape of the seed, color of the seed, color of seed coat. So in the plant height, tall and dwarf, tall is dominant and dwarf is recessive. So like that he has studied. So you can see in the table. So which is dominant and which is recessive. Again, I'm not reading and because to save the time. Okay, you can just go through that. And now gene symbols. So symbols are given as by English alphabets or letters. So here usually the dominant is given by the capital letter and recessive is given with a small letter. Okay. Then homozygous and heterozygous. So homozygous means two alleles of in the homologous chromosomes, that is usually they'll have two copies of chromosomes, same chromosome, like first chromosome two copies, second chromosome two copies, like that. So when the two alleles are present, if the two alleles are of same kind, whether it is dominant or recessive, okay, if it is dominant, capital A, if it is small recessive, small a, like that, they are called homozygous alleles. Okay. And they produce only one type of gamete, that is capital. If suppose capital A is present, it will definitely give capital A gamete. Okay. And such individuals are called pure breeding and they produce same type of individuals when they are selfed. And only one type of class is observed when we see this. Then heterozygous means dissimilar alleles are present. That is having dissimilar genes or alleles, like capital A, small a, as I told you, capital T, small t, like that. They produce two types of gametes. It can give capital A or small a. And they produce three types of offsprings. So the offsprings which are having capital A, small a, and capital A, small a. Again after selfing. So these three different kinds are observed in second filial generation or F2 generation. Okay. Then pure line. So a pure line is a plant which breeds true on selfing. Means they produce only one type of offspring on self-pollination. Okay. That is Mendel developed pure lines of purple and white color, yellow and green color, seed color, and round and wrinkled seed shape, like that. All the characters he took only pure lines. Then crossing over. So artificial pollination between plants which give contrasting characters. That is called crossing over. That means different, like hybrid kind of characters are generated here. So this is called F1 generation, in F1 first filial generation. And depending on the gene pair involved, crosses are monohybrid cross, dihybrid cross, and polyhybrid cross. So even like this is the experimentation, but in the nature, usually we see a crossing over. Okay, because we have variation, even we don't look similar, exactly similar as that of our parents, right? So we'll have some kind of variation. Then Mendel first selected homozygous plants, which are called parental plants, and P generation. Then after crossing, whatever the generation he got, that is called F1 generation or first filial generation. Then in the second, after crossing them, self-crossing or cross-fertilization, then we'll get second filial generation or F2 generation. Okay. Now here, first we will discuss about monohybrid experimentation. So I will just explain you this image so that will be easy for you. So here parental generation. So it's a pure breeding tall and pure breeding dwarf. Capital T and small t, small t. So capital T, now these capital T, there are two copies. They will get separated into gametes. That is whether it is, suppose if it is like male or female gamete, whatever it is. So you have two gametes, capital T, egg cells. Then here in the dwarf, small t, they are also separated into small t, which are separated as pollen, male gametes. Now crossing over takes place. Now whatever way they cross over, so we get two individuals in F1 or first filial generation. They are capital T, small t. They are having, okay, because one from the egg cell and will fertilize with the one pollen, right? One egg cell fuses with or fertilizes with the one pollen. So one egg, whatever egg comes, it is having capital T. And whatever the pollen coming from the male side, it is having small t. So, individual in the first generation will definitely get capital T, small t. But the phenotype is tall only. Okay, because capital T is dominant over small t. Now, again in the next generation, when this capital T, small t is, that individual or that egg cell is fused with other individual. Now capital T, small t will give the gametes as capital T, one egg cell, small t, another egg cell. In the same way, capital T in one pollen and small t in another pollen. Now when they fuse, when one egg fuses with the pollen, the egg may have capital T or may have small t. In the same way, the pollen also may have capital T or may have small t. Now, when the fusion takes place, there are four possibilities. So, the individual may get capital T, or capital T, or small t, right? When he observed in the ratio, like it is not that when you see plants, it is not only one individual, like in human beings, only one child or two children, like that. In plants, any number of plants they may develop. So accordingly, when he observed, he found a ratio. So genotypic ratio is nothing but one tall homozyte, that is capital T. There are two, here it is given as 3, but it is 2. Two heterozygotes, that is capital T, small t, and one dwarf, that is small t, capital T 1, capital T, small t 2, small t 1. So 1:2:1 is the genotypic ratio. Whereas in the phenotypic ratio, it is pretty tall, because whether it is capital T or capital T, small t, it is tall only, right? So three individuals, suppose if there are four individuals, three individuals will be tall one individual will be dwarf. Okay. So you have to please correct it. It is in the genotypic ratio 1:2:1. It is not 1:3:1. Okay. So this ratio he was observed. So what is this? Phenotypic ratio is 3:1. Genotypic ratio is 1:2:1. So this is called monohybrid ratio. So mostly in most of the organisms, whatever he found, he got same ratio. Like even after that, even now for human beings also, if they perform this experiment, like performance theoretically, when they see, so they should get same kind of ratio. Okay, there are some exceptions also. We will discuss that also. So this we can actually develop by checkerboards. So whatever they represented here, this colored box, checker box, which is called as Punnett square, because this was developed or introduced by R.C. Punnett in 1906. Okay. Then you have dihybrid experiments. So after dihybrid experiment, I'll stop. So you just practice today once. Same example you take from the textbook and you practice once. If you, although I'm telling it theoretically, if you practice only, you'll observe, you will understand. And if you find anything difficult, you just let me know. So because these are very important topics. Okay. So in the dihybrid cross, so in the monohybrid, we took only one character, tall. Okay. Only tall and dwarf. Only, but height is only one character. But here he took two characters at a time. See, in human beings, for example, for your understanding, I'm telling. So we cannot just study only one character at a time. Only hair color, or hair style, type of hair, or height of a person, or color of a person. We cannot be, there is nothing which comes out if we study individual characters, because the genes in real, they'll be interacting with each other. One depends on the other. So you cannot study each gene individually. That is only for your study purpose. But in real, it should be dihybrid or trihybrid cross. So in the dihybrid experimentation, if you see, so he took two characters. So one parent is having capital R, capital Y, which is round yellow. Then another individual is having smaller, small y, but which is wrinkled green. So it is the shape of the seed and color of the seed. Okay. When they performed cross between these two plants, so this parent with capital Y will give the gamete, which is capital R, capital Y. Okay. And in the other individual, it is giving small r, small y. Now, when these two are crossed in the F1 generation, they'll get capital R, small r, capital Y, small y. This is the genotype. But the phenotype will be round and yellow, because capital R is dominant and capital Y is dominant. Okay. Now, this capital R, capital Y, small y, when it is self-fertilized, that means another individual is also having capital R, capital Y, small y. Now, when these two are fused or fertilized, crossed, you will get different number of combinations. Total 16 combinations you will get. Here you can see. Now, how the gametes will be? Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Okay. Here, other individual also you'll have 4 types. Okay. Now you have to combine these two. So first box you see capital R, capital Y, capital R, capital Y. Both, if you combine, what you'll get? Capital R, capital R, capital Y, capital Y. Okay. Then second box, capital R, small r, capital Y, capital Y. Capital R, small y, capital Y, small y. Like that, you have to do combinations and you have to select the genotype and phenotype ratios. So, in the genotype, usually it will be very big. if you have to calculate. But in phenotype, usually capital R, capital Y means round yellow. So here, usually yellow round will be 9, yellow wrinkle will be 3, green round will be 3, and green wrinkle will be 1. 9:3:3:1 is the ratio you will get in the phenotype. In the genotype, you will see 1:2:2:1:4:1:2:3:1. Like that you will get. So what I will do is homework. Okay, I will other classes all I will tell tomorrow. What you have to do is do practice monohybrid and dihybrid cross. Same experiment you take from the textbook. And if you understand this only, you can go to the next topics. You will understand easily. Is it clear? Any doubts still here?
No, ma'am.
Okay. So once you do this, tomorrow we will discuss the next topics. Okay. If possible, discuss with your friends also. Any topics you want special attention, you can just tell me. Okay.
Yes, ma'am. Thank you.
Okay.